I'm pleased to be here to speak to you. I heard about the Institute for Continued Learning and never had the chance. I was never invited. And then finally one day I got invited and I graciously accepted. And I admire the ICL because I believe in the curious mind and the mind that remains curious throughout one's life, not just the 4 years that you're in college. As Don alluded to, I am Canadian. I apologize for that. No, we're always apologizing, so it's a big joke. But to give you a little more in-depth background, I was born in Vancouver Island, and my father was in the military, so by the time I graduated from high school, I had lived in every province in the country, all 10, which was both a blessing and a curse. I attended the University of Manitoba, where I got a bachelor's degree in zoology, and my interest became more focused to physiology. So I enrolled in a graduate program in physiology, and after 2 years, my committee said, "We want you to continue pursuing your PhD," and I said, "I'm not going to do that. I'm going to graduate with a master's degree." I was not satisfied with studying organisms at the level of physiology. It was sort of a black box. You treat an organism with drugs and you measure what comes out the other side, and something is happening inside, but you don't quite know what it is. And that was not satisfactory to me. I wanted to look deeper, so I enrolled in a PhD program at the University of Southern California, where I studied molecular biology. So looking at the inner workings of a cell, not a tissue or not an organism, but looking deep inside. And that was deeply satisfying. After my PhD at USC, I went to the University of California, San Diego, where I continued my studies, looking at the cytoskeleton, a component of the cell, in the lab of Dr. Larry Goldstein, who was a Howard Hughes Medical Institute fellow, so it was a very large lab. And while the research was satisfying, the environment was not. It was deeply impersonal. I think there was something like 30 graduate students in the lab, and I would get 5 minutes of my advisor's time a week. And they were meaningful 5 minutes, but they were not satisfying. So when it came time to looking for a permanent position, I knew that I wanted to attend an institution where I had the best of both worlds, that I could continue doing research while I taught, but that I could engage with students while I was doing it. And so here I am while I'm at university. What Don has asked me to do is to talk about DNA and human evolution. My understanding is that you are learning components of this course from Dr. David Sadava, understanding genetics, DNA, genes, and their real-world applications. And I was asked to provide some insight on DNA and human evolution. And I asked Don, do I have the entire semester to do this? And he said, no, you have 2 hours. And I said, well, okay, it'll have to be condensed, it'll have to be precise, but I think I have materials here that will enlighten you and are sort of on par or on spirit with this course. I did look online and see that this was produced in 2008, almost 10 years ago. Molecular genetics is probably the fastest, most rapidly growing field in biology. So 10 years is a long period of time. Let me give you an example. I'll give you 2 examples. When I started my PhD in 1995, my first experiment was to clone a gene from the fruit fly Drosophila. That took me almost an entire year to do. By the time I graduated 6 years later, it took 3 minutes to clone a gene in Drosophila because all you had to do was pick up the phone and call a company and tell them what gene you wanted, have your credit card in hand, give them their number, and it was in the mail to you 2 days later. So this field evolves incredibly rapidly. The second example, which you're probably more familiar with, is the Human Genome Project. So the Human Genome Project was instituted or started by the Department of Energy, and their goal was to sequence all 3.5 billion base pairs of a human's genome or DNA. That took 15 years, probably 8 or 10 different countries involved, with hundreds, if not thousands of scientists at a cost of $3.5 billion. 10 years later, sequencing a human genome takes one individual in one room with a machine about the size of a photocopier. It takes 1 day, and it costs around $10,000. So this field is, and the goal is to do it for $1,000, and that is absolutely attainable. So this field is changing incredibly rapidly. So as good as Sadava's lectures are, there are things that have changed, things that have not changed. The structure of DNA is still the same as it was 10 years ago, but more recent advances have been made, and there are better examples of human evolution, and I'm going to touch on those. So my plan for today is that I will present material in sort of 3 parts. The first part, I'm going to answer the question, what is DNA? So it's essentially ubiquitous in our society. You hear about it all the time, you see pictures of it, it's in movies, everywhere, but what is it? And importantly, how does its structure? So understanding its structure is critical, because if you can understand the structure of DNA, then it's very easy to understand its 2 major functions, and that is to act as a molecule that stores information and to act as a molecule that can transfer that information from generation to generation to generation. So the first thing we're going to do is look at DNA at a structural level. The second thing we're going to do is, what is evolution? Try and answer that question. Now, I am not an evolutionary biologist. I am a cell biologist and a geneticist, so I do know enough about evolution, just enough to get me into trouble. So we will explore what is evolution, in particular, what is natural selection, and what is adaptation. These 2 things are very important for understanding the third part, which is evidence for human evolution. What evidence exists that humans themselves are not only the product of evolution, but are still subject to evolutionary changes? And in between, we'll take time for questions and a break. I don't know what the, Don, what is the standard? 10-minute break? Okay, great. So what is DNA? DNA is a very complex molecule, but the good news is it's built from a very simple molecule. So if you can understand the simple molecule, DNA is just built from a whole bunch of simple molecules. So the simple molecule may not look so simple to some of you, but I'll try and make it as simple as possible. It's called a nucleotide, and it has 3 basic parts that are color-coded. It has a sugar, right here in blue, a ribose sugar, and attached to that ribose sugar is a phosphate, and then this more complex structure called a nitrogenous base. So the acronym DNA stands for deoxyribose. Why? Because the ribose lacks an oxygen right here. There should be one, but it lacks it. So deoxyribose, nucleic acid. So DNA is deoxyribose nucleic acid. This is the basic building block. So DNA is just a whole bunch of these joined together. There are 4 basic types of nucleotide. and they differ from one another on the nitrogenous base. So all nucleotides have the same ribose sugar, they have the same phosphate, but they're different from one another on this nitrogenous base. So the nucleotide adenine has this structure as its nitrogenous base. The nucleotide thymine is different from adenine in that the nitrogenous base has a different structure. Guanine has a different structure than thymine. It's very similar to adenine, but it is different. And then you have cytosine. So there are 4 nucleotides that are joined together to create a DNA molecule. All the nucleotides share the same feature of a sugar and a phosphate, and they differ from one another in these nitrogenous bases. So these are routinely known as A, G, T, and C by their abbreviations. But A is adenine, T is thymine, G is guanine, and C is cytosine. So these are the building blocks of DNA. And if you look at these molecules have polarity. So what does polarity mean? This marker has polarity. You can identify one end from the other because it is different. One end has a cap, the other end lacks a cap. So nucleotides, they also have polarity, and the polarity is right here. So one end of the nucleotide has a sugar, the other end of the nucleotide has a phosphate. Okay, so these building blocks, they have polarity in much the same way this marker does. So those molecules are called monomers. They're very small subunits, and if you want to build a polymer, poly meaning many, you simply join monomers together, like beads in a necklace. So you can make a polymer of nucleotides by joining them. The phosphate of one nucleotide is attached to the sugar of the other in sort of a head-to-tail arrangement. And if you keep doing this, you build a polymer. But because the individual molecules have polarity, and because you're joining them head to tail, you can build an incredibly long polymer. But that polymer has polarity as well. One end has the cap, the other end lacks the cap. So if you have a monomer which has polarity and you join them head to tail, you will build a polymer that also has polarity. This is a common way that complex molecules are built in biology. They're built from very simple molecules. So as 2 scientists, James Watson and Francis Crick, Watson was an American and Crick was a Brit, as they tried to elucidate the structure of DNA, they were working with information that was presented to them by other scientists, willingly or unwillingly. That's a long story. But one of the pieces of information that Watson and Crick had was that another scientist, Erwin Chargaff, he would isolate DNA from all sorts of organisms. Bacteria, salmon, chickens, humans, it didn't matter. Whatever organism he isolated DNA from, when he analyzed that DNA, he always found that the percentage of guanine that was present in DNA was exactly the same as the amount of cytosine, and that the amount of thymine that was present in DNA was always exactly the same as the amount that was present with adenine. These were called Chargaff's equivalencies, and they helped inform Watson and Crick what the actual structure of DNA was. Because Watson and Crick were model builders. They used information gathered by other scientists to actually try and build physical models that correlated with the data that was available at the time. And so they knew that DNA was comprised of nucleotides, that nucleotides could be joined end to end, and that the percentage of guanine was equivalent to cytosine, and the percentage of thymine was equivalent to adenine, always in the DNA from every organism. So they started building physical models to try and determine what the structure of DNA was, and that involved making physical representations of the DNA. So here's James Watson in his senior years describing exactly how he came to devise what the actual structure of DNA was.
And moving them around, and one right on top of the other. One molecule, all fitting into this wonderful symmetry which we saw, the morning of February 28th, 1953.
So I'm pretty sure that's not exactly how it happened. It's a nice representation, but what Watson and Crick discovered was that DNA is actually made of two polynucleotide chains. So two chains that are assembled of nucleotides, head to tail, and that the base pairs in each chain interacted with each other. So the reason why Chargaff observed that the amount of thymine was always the same as the amount of adenine is because they physically bonded with each other. And the amount of cytosine was always the same as guanine because they physically bonded with each other. And that DNA wasn't comprised of a single chain of polynucleotides, but rather it was comprised of two chains. And the two chains, get my polynucleotide here, the two chains were actually anti-parallel, so they ran in opposite directions. And the bases from the chains stuck out inwards towards the center and interacted with each other. Adenine always base pairing with thymine, cytosine always base pairing with guanine. And this was an amazing revelation because immediately the structure of DNA immediately suggested two things: how DNA could be transferred faithfully and how you could store information in that molecule, all in one structure. So this is what DNA looks like. The orange here, that is the sugar and the phosphates connected to each other. That is the backbone of the molecule. And the nitrogenous bases, they stick inwards towards the center. It has the exact same structure as a ladder. It just happens to be a spiral ladder. And the steps of the ladder are the bases that stick inwards, and the sides of the ladder that support the steps, that represents the sugars and the phosphates repeating over and over again. So if you look at this molecule, it is DNA. It is two polynucleotide chains that are anti-parallel with the nitrogenous bases facing inwards, interacting with each other. Adenine always with thymine, guanine always with cytosine. And I would like you to note that it is a right-handed helix. It is a double helix. There are two chains here. That is one chain, and there's the second chain. So are there any questions at this point about the structure of DNA? The one at the, yes, it is significant. So cytosine and guanine, they are linked to each other by three bonds. They're called hydrogen bonds. They're weak chemical bonds. Thymine and adenine are connected to each other by 2 hydrogen bonds. There is almost the possibility of having a hydrogen bond there, but the distance is just too far, so it doesn't happen. So technically speaking, cytosine and guanine are harder to separate from each other than adenine and thymine are. So the linkages between these are called hydrogen bonds. They're very weak chemical bonds.
What triggers the separation?
Okay, so what triggers a separation? In a lab, if you have DNA in a solution, you can heat it to boiling, 100 degrees, and that will separate both strands. And if you allow it to cool slowly, the strands will find their proper partners and reanneal perfectly. In a biological context, there are enzymes that will separate the strands when the strands need to be separated. And that is typically during a process we call replication, when the DNA has to be copied. Why? I'm not sure why right-handed, but I know that it is not left-handed. And there is no shortage of examples of left-handed DNA. So one of the things that I teach in my class is I make sure students understand absolutely that DNA is a right-handed helix. So what is a right-handed helix? If you take your right hand, and you face the molecule and you stick your thumb up, your thumb represents the axis of the molecule. Your fingers will indicate the direction that the backbone should be coiling. So it should be coiling in that direction as you go from top to bottom, which it is. Now, it doesn't make much sense because if a staircase is a right-handed helix, as you go up the stairs, you are constantly making left turns. So it can be confusing, but a right-handed helix, you take your right hand, it points up, your fingers are wrapping this way, so as the helix goes up, you should be going in the direction of your fingers. Now, if you find that this is confusing, you are not alone. Time, Newsweek, common magazines always publish DNA with the incorrect coiling. Postage stamps issued even by my native Canada incorrectly. Those do not exist in biology. Or they do, but they're very temporary moments of small regions that become left-handed because proteins twist them. Even biological textbooks. The other night, I was watching Jurassic World, the new installation of the Jurassic series, where a scientist is explaining this new genetic variant of a dinosaur that they made. And what happened? The dinosaur got free and went wild and ate a whole bunch of people. I have a theory for why that happened. So Indominus Rex, I understand now why it went wild and ate everybody in the park. It was synthesized from left-handed DNA. I'm sure if they had used right-handed DNA, there would not have been a problem. And I should say, my poor wife, I made her rewind this movie like 3 times so I could actually video that and display it in class. But I emphasize this point so much to my students in gene structure and function, a 300-level course I teach, that years later, I receive emails from students saying, I'm at a scientific meeting and Dr. So-and-so is giving a presentation, and they actually took a picture of him on stage with his presentation, and lo and behold, there's a left-handed helix. And so in the instructional lab on campus, we have a left-handed Hall of Shame where we post pictures of of all of these. And it's very difficult to identify. I mean, even Dr. Sadava of the Great Lectures was not immune to this. He delivered all 24 of his lectures standing in front of a left-handed helix. So, to be certain, left-handed DNA does not exist in biology. It's a right-handed helix. So, Francis Crick, James Watson, and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962 for their discoveries concerning the molecular structure of nucleic acids, or DNA, but most importantly, its significance for information transfer. Because as I said, as soon as this structure was elucidated, it became immediately clear how this molecule could serve its two functions: information storage and information transfer. This is Max Perutz and Kendrew. They won the Nobel Prize in Chemistry for their study of proteins. Anybody have any idea who this person is? Yep, it is John Steinbeck, American author John Steinbeck, who won the Nobel Prize in Literature in 1962 as well. That I did not know until I prepared this lecture, but that is John Steinbeck, cushion between Crick and Watson. Okay, so how does the structure of DNA, how does that amazing, beautiful molecule facilitate its two main functions, which I've told you are it's the storage of information and the transfer of information. And when I mean transfer of information, I mean from generation to generation to generation. So in their one-page publication that essentially won them the Nobel Prize, so anytime I give students an assignment that is 1 page long and they complain that that's just not enough room, I tell them, well, it was enough room for Watson and Crick. They won a Nobel Prize with it. In their publication in Nature, this paragraph here states, "It has not escaped our notice that the specific pairing that we suggest, which is adenine is always base pairing with thymine and cytosine with guanine, so A and T are always together, G and C, that this pairing suggests a possible copying mechanism. And the copying mechanism is simple and it is elegant. It is, all you have to do is take the parental DNA, which is blue, and split it, and each strand of the parent DNA can act as a template for the synthesis of the other strand. So a molecule can come around and say, well, there's a T here, and I know that adenine always pairs with T. There's a guanine here, cytosine always pairs. Thymine, adenine. So you can build this strand. You can determine the order of nucleotides that should be put into the polynucleotide chain by simply reading what is on that strand. Incredibly elegant, and it turned out to be true. It just took 30 years to determine the exact mechanism of how that happens. So how about information storage? How do you store information in DNA? So here's DNA. You know it to be double-stranded, and it's comprised of 4 nucleotides: guanine, cytosine, adenine, and thymine. And here they're green, red, yellow, and blue. DNA's function is that it contains information that results in the synthesis of a protein. So what is a protein? A protein is a complex molecule that is built from simple molecules, just like DNA. But the molecules are different. The molecules are called amino acids, and instead of 4 of them, there are 20 amino acids. So it is essentially the equivalent of a pearl necklace, but you have 20 different types of pearls that you can assemble into your necklace. So how does the information that's contained in DNA direct the synthesis or the order that amino acids should be synthesized in a polypeptide chain? So it could be that it's a one base code. So an adenine says you need to put amino acid number 1. Thymine, a different amino acid number 2. Guanine, amino acid number 3. Cytosine, amino acid number 4. But what is the problem with that? There are more amino acids than there are possible combinations or codes. So maybe it's a 2-base code. Well, a 2-base code gives you 16 different combinations. You have AA, AT, AG, AC. Please don't make me go through all of them. Trust me, there are 16 of them. And the problem again is not enough. You don't have enough information to specify all 20 amino acids. So it became very clear that the genetic code is likely a 3-base code. In a 3-base code, you can have 3 adjoining nucleotides in 64 different combinations, 64 possible combinations. And that's more than enough to specify the order of all the amino acids. It actually allows for some redundancy. So the genetic code that is stored in DNA is a 3-base code system. This was literally predicted the day after the discovery of the structure of DNA, and again, was proven some 20 years later. So a code of 3 consecutive nucleotides was the only code that was sufficient to specify all 20 amino acids with some redundancy built in. Some amino acids are very lucky. They're specified by AAA, as well as AAT, as well as AAG, as well as AAC. And some amino acids are not so lucky. They're specified by only one codon. So it turned out, as predicted, the linear order of nucleotides, so how the order or the sequence of the nucleotides in a DNA chain, that is codons, determines or encodes the linear order of amino acids in a polypeptide chain. So not only was this molecule capable of storing information, but that information could be transferred generationally. Generation after generation after generation. Now the hitch in all of this is that the transfer of that information is not always faithful. And that is the basis for evolution. So if you have a change in a nucleotide, so this codon here is ATG, okay? If that was to change, now it's ATA, that might specify a different amino acid be incorporated in that chain. And why is that important? Well, because proteins are the molecules that do work in a cell. And if evolutionary change happens at the level of the DNA, evolutionary pressure or selection, that occurs at the level of the polypeptide, at the level of the protein. So if a change occurs in the DNA, an organism pays the consequence with what that change does to the protein and ultimately what that protein does to the health of the organism. It might make the organism better, in which case it might be selected for in the population and be maintained, or it might make the organism sicker, in which case it would be selected against. So the movie that I'm about to show you details what I think is probably the most brilliant example of an evolutionary change that occurs at the level of the DNA and how it resulted in the change of a protein that benefited an organism and became fixed in the population. And that is, the mutation occurs at the level of DNA, and it's random. Sometimes it happens in this gene or that gene, but for that one dark mouse, it was a great mutation if it lives close to or on that black terrain, because it stands a significantly better chance of reproducing and passing that trait on. If the converse happened, if a mouse who lives in the desert had a mutation that occurred in the same gene, but it made that mouse's fur black, that mouse would probably be eaten very quickly and easily, and the chance of it passing its genes on, essentially nil. So, two things to take away from that. Mutation, it occurs randomly, and it is just a part of being a biological organism. But whether that mutation is, quote, good or, quote, bad, depends on the evolutionary pressure that organism experiences. If a polar bear has a mutation in its gene that produces pigment and suddenly it's black, that is not a favorable phenotype or mutation to have in the Arctic, where you're trying to sneak up on prey in, well, at least what was once covered in snow. Maybe it will be a favorable mutation in the future, who knows? So this is where evolutionary change happens, but at the level of the protein, the function of the cell, the function of the organism, that's where evolutionary pressure is realized. And whether a mutation becomes a permanent part of a population and spreads rapidly, or whether it is extinguished. So there are significantly greater mutations that happen and are unsuccessful and are extinguished before we ever know about it. All right, so are there any questions on either DNA structure or evolution? Yep, okay, so the question is, I'm using genetic code and gene sort of interchangeably. So every single one of you has, in every one of your cells, except gametes, you have 2 copies of what is called a genome. You inherit 1 copy from mom and 1 copy from dad. That genome is 23 chromosomes, and each chromosome is one long giant strand of DNA. Embedded in that DNA are genes, segments of that long chain that will actually make a protein. So truth be told, of those 3.5 billion base pairs that make up your genetic information, only 2% actually makes a gene. So the obvious question is, what is the other 98% doing? A large portion of that is involved in controlling those genes, ensuring that they're turned on in the right cells, at the right time, in the right places. So the genetic code, that refers to essentially the code that allows you to decipher a gene. It's essentially a piece of paper that you can look at and know that AAT specifies the amino acid alanine, and GAG specifies a different amino acid. So it's the translatable code. A gene is a small segment of a large polynucleotide chain, or a chromosome, that actually encodes information to make a protein from start to finish. Does that answer your question?
Jeanette, thank you so much. I was interested in this right-handed helix as
Yep.
opposed to the left hand. I mean, if the scientists disagree on which way it goes. Does it matter?
So there's no disagreement. It is a right-handed helix. Why it is a right-handed helix, I do not know. I suppose it's maybe has a more favorable conformation. I think that might be something for maybe an organic chemist. Or I do not know why. But the reason why you see so many left-handed helices, there's a one-word answer for that: Photoshop. So most of those things like postage stamps and the covers of books, those are not designed by scientists. Those are designed by graphic artists. And so what they do is they either design a structure incorrectly, there's a 50% chance that they do that, or if they have a correct structure, as they're designing something, they flip horizontal. So you can take a correctly oriented DNA, you can rotate it all you want, you can even flip it vertically, no problem. But as soon as you flip it horizontal, you've made an incorrect DNA molecule. So there's no disagreement. I just marvel at how often the structure is misrepresented. And I focus on that in one of my classes because it really gets my students' attention focused. They think about it. I actually have them design a DNA molecule out of clay. We have these glass tubes, and they wrap the clay strands around the glass tube and make bases and connect it. I actually have them physically construct it so they make it with their hands. So they get that tactile connection with what it means to be a right-handed helix. For those of you that don't know, I arrived to Willamette in 2008.
How do you explain more complex evolutionary changes? For example, for a bacteria going from having a tail to having a flagellum.
Yep.
That means that there has to be motility, proteins synthesized, several other structural proteins synthesized, and there are no intermediary forms that are ever seen.
Yep.
So how do you explain that one big step that occurs as evolutionary?
Yep, so, I mean, there's a lot of examples of this in biology. How do you explain the evolution of the eye? Something so complex. How can you explain that? Was it that an organism, didn't have an eye and then, boom, there was a mutation in a single gene that, bang, caused it to develop an eye? And clearly it's not that simple, but it is small changes that occur and maybe produce intermediate forms that we're unaware of, that there's no fossil record for because do you want to ask it? Go ahead. I can't ask. Oh, you're not allowed? Is that a rule? Is that a rule? Okay. So I will agree with you. It is hard to comprehend how humans could have evolved. Look at us. I mean, look at us. We're humans. So I'll tell you a little story. When the human genome was sequenced, there was this rogue scientist who said, my company will sequence the human genome. We're going to do it in 4 years. This was before the government effort had finished. And he said, as a test run, we're going to sequence the genome of the fruit fly in 8 months. And lo and behold, they did it. They sequenced the genome of the fruit fly in 8 months. And it turned out the fruit fly has 13,500 genes. And at that time, scientists were saying, well, look at the fruit fly and look at us. We're way more complex than a fruit fly. Estimates for human genomes were that we must have 250, maybe 500,000 genes. Look how complex we are. Do you know how many genes there are in the human genome? 35,000. 35,000 genes. So we're 3 times more complex than a fruit fly. If you look at the level of the gene. But as it turns out, we're much more creative in how we use our genes. We can use our genes, I'm not going to get into details, but we can have a single gene and we can make many, many different proteins from that gene. And it's based on the way our genes are organized. They are organized not like a single continuous story, but they are organized almost in chapters. And if you were a child and you ever had those books, they're called Make My Own Story books. It was one book, but you'd read the first chapter and then, there'd be a question at the end. If Joni should kiss Chachi, go to page 62. If Joni doesn't kiss Chachi, go to page 45. And you could create your own story by piecing together different chapters. That's what humans do. So we don't have 500,000 genes. That discovery was a blow to the ego of many geneticists. But we use our genes to make use our 35,000 genes to make 500,000 proteins. So part of the answer to your question is that there's tremendous complexity in us, and a single change in the DNA can often result in a dramatic change in the appearance or the phenotype of an organism.
Hi, my name is Franca. The question I had to do a lot with mutations. And so I'm seeing a little bit of the randomness of the mutations. Is it possible that living organisms contain latent mutations that are there ready to be utilized at the right moment, and then is it possible the mice could go back to being white if the right conditions presented themselves?
It is possible that you could have a black mouse could incur a second mutation. So here's a great example. You have a black mouse, you have a white mouse. That's the natural state. And in order to make pigment, it requires two genes. One gene to actually synthesize and make the pigment, and then the second gene to actually transport the pigment into the hairs. Okay? So that white mouse has a mutation in a gene which turns it black. Why? Because the gene that makes the protein pigment has been mutated, so it makes the pigment and it's transported into the hair cell. So now that mouse that used to be white is black because of the first mutation. Generations, one of its future progeny has a mutation in the gene that transports that pigment. So like its great-great-great-great-grandfather, it is now white. So you can have multiple mutations in different genes that affect organisms in randomly. So the whole idea of this is that mutations are random, they happen. I should make a bumper sticker. Mutations happen. They happen. They're a part of being a biological organism. There's no disputing that they happen. The question is, what is the consequence on the organism when they do happen? And for that mouse that was mutated, or had a mutation in a gene that went from making it gray to making it black, it found a new environment that was hospitable to it, that black molten rock in New Mexico. And once it got there, it reproduced and proliferated like crazy, transferring that mutation to all of its progeny.
We have time for one last question.
Did someone say why? Because it had a leg up on all its competitors. Because it was hidden from its... The evolutionary pressure is the predators that are feeding on the prey. They are more likely to feed on the prey that they can see than the ones that are camouflaged. And so you know what happens? Is that mutations also occur in the genes of the predators. So suddenly, a predator has a mutation in a gene that changes its eye so it can discriminate color better. So it becomes a better predator. And what happens? the prey become better prey, more concealed. And it's almost like an evolutionary arms race between the predator and the prey. That is called coevolution. And there are lots of examples of that. It's called coevolution because both the predator and the prey are evolving in a way that affects each other. It's almost like a tug of war. You can think of it as a genetic tug of war that happens over hundreds, if not thousands of years.
Have they identified certain things or procedures that turn the code on or off? And then also, why do some mutations never seem to disappear of genes?
You mean from a population?
Yeah.
Or from a...
From the population.
So recall that when I said, you have 3.5 billion base pairs, and only 2% of that is, quote, important for making a protein. If you have a mutation in a segment of DNA that doesn't encode a protein, who cares, right? There's no evolutionary pressure for keeping it or losing it. In an evolutionary context, the only mutations that are important are going to be the ones that ultimately affect the fitness of an individual or the proteins that are made. And so we accumulate, as humans, we accumulate mutations in our DNA all the time. All right, everyone, welcome back. I've got a lot of questions about right-handed and left-handed DNA. I would like to apologize to anybody who's left-handed out there. I don't mean to offend, but one of the great questions, and ashamedly, I don't know the answer, why right-handed versus left-handed? And I do not know the answer. I will question Dr. Google when I get back to my office and try and find out why, but I am not sure why a right-handed helix is the preferred orientation. But I was reminded of the importance between left-handedness and right-handedness when you're thinking about molecules, and the example that was brought forward was thalidomide. So thalidomide was a drug that was synthesized for one purpose, and it ended up causing severe problems, birth defects in mothers who took this drug while they were pregnant, and ultimately it was discovered that the reason why is because the form of thalidomide that they were given had a mixture of the exact same molecule in its right-handed form and its left-handed form, and it was one form was therapeutic and the other form caused severe problems and birth defects. So handedness, at least in a molecular or biological context, is important. All right, so I thought I would follow up with this sort of introduction of the structure of DNA, understanding that embedded in DNA is a code, and that code changes. I think an important thing to understand is mutation happens. Mutation is natural. Mutation is actually a consequence of copying your DNA. So every time you cut yourself and that wound starts to heal because cells are dividing, every time a cell divides, that cell is asked to copy 3.5 billion base pairs of DNA sequence, not once, twice, and pass one copy on to each of the daughter cells. You're going to make some mistakes when you do that. And the mistakes are rare. They are infrequent. I think the order is 40 to 50 mutations per 3.5 billion base pairs. I don't know what that percentage is, but it's probably 99.9999% accurate. On top of that, mutation in roughly only 2% of that is going to affect the organism, so we live inconsequential of those mistakes. But it's important to understand that they happen. It's a natural part of an individual's biology. Mutations can be introduced in the DNA in lots of other ways. Chemical teratogens and X-rays and all sorts of other things, they do the trick too. But just being a biological organism does it as well. And I've heard people say a lot, Mother Nature is perfect. But in a biological context, Mother Nature is perfect because she is slightly imperfect. That's what makes her perfect. So when she copies the DNA, she makes mistakes. Those mistakes are inherited, and those mistakes result in change in the form of organisms over long periods of time. So there's another bumper sticker. Mother Nature's perfection is she is slightly imperfect. All right, so how about humans and human evolution? So in his lecture, Sadava talks about probably the most common example of human evolution, and that is sickle cell disease. So I'm going to touch on that. And then I'm going to touch on an example that he doesn't mention. And I like this second example. It's called lactase persistence. And the reason why I like it is because it's an example of an evolutionary event that has happened twice in the human population independently. And it is evolution that is driven by a selective pressure, which is not a predator or anything like that, but rather it is driven by culture, how an organism interacts with its environment. Okay, so sickle cell disease. So coursing through your blood right now, I think it's on the order of 5 liters or so, are these cells called red blood cells. So they're disc-shaped, they're shaped like donuts. They contain inside of them a protein called hemoglobin. Now, because hemoglobin is a protein, that means in your DNA there is a gene that directs the synthesis of that protein. So there's a sequence in your DNA that's responsible for making hemoglobin. Hemoglobin is actually made up of 2 different proteins, an alpha subunit and a beta subunit joined together. So there are 2 different genes responsible for making hemoglobin. What is the function of hemoglobin? It binds oxygen, it carries it to the tissue, and when it gets there, it releases it. So if you look at the sequence, and not the entire sequence, but only part of the sequence of the normal, I don't like to use normal, of the wild type beta globin gene. So here's the DNA sequence. It's only a short segment or part of it. TGA, that is a sequence of nucleotides that makes the amino acid, or directs the amino acid threonine to be incorporated in the protein. GGA, proline. CTC, glutamic acid. CTC again, glutamic acid, so on. That results in hemoglobin being made that is normal and red blood cells being made that are normal. There is a mutation, a change at the level of the DNA that occurs in some individuals that changes this thymine to an adenine. So this code, CTC, has been changed to CAC. What is the consequence? The consequence is that when this individual builds a protein for beta globin, one of the subunits of hemoglobin, instead of incorporating a glutamic acid after that proline, it incorporates a valine. So beta globin, I think, is 120 amino acids long, and in this individual, 119 of them are correct. This one is incorrect. It doesn't seem like a big difference, right? more than 99% of the amino acids are correct. Only one amino acid is incorrect. But the consequences on the organism are tremendous. This individual will produce hemoglobin that is not normal, that instead binds to all the other hemoglobin that has the same mutation and forms these long strings of hemoglobin, these long hemoglobin rods. And what does that do? It changes the shape of the red blood cell so that it becomes sickled, shaped like a sickle. So one small change at the level of the DNA, tiny, results in a huge impact on the organism, a huge physiological impact. So a scientist named A.C. Allison asked a very simple question. And his question was, if this mutation causes a different form of that protein to be formed and messes up your hemoglobin and causes the red blood cells to be misshapen, and individuals who have this, or 2 copies of this, to have sickness and illness and disease and pain, why does it persist in the population? This seems like it would be a bad thing to have happen. If it's bad, then why does it exist in the population? And so this mutation that affected hemoglobin protein was discovered in Africans. And what A.C. Allison did is he looked at a geographic distribution of where that mutation occurred in individuals. Where were they from? And so that's what's represented here. So mutations in that gene, individuals that have that mutation, are predominantly from these regions of Africa. Less so in these regions here. And he looked at that geographical distribution and he said, my, that looks quite similar to the distribution of malaria. So malaria is a parasite that has a life cycle that begins in a small organism called a tsetse fly. And what does it do? The tsetse fly bites a human and it transmits the malaria to the bloodstream, where it goes into red blood cells, reproduces, makes the individual sick, but along comes another tsetse fly without the infection, bites the human, draws some of the malarial cohorts out of the blood, and flies over and infects another individual. So humans are an intermediate host to malaria. The distribution of malaria and where it occurs sharply overlapped with the distribution of where this mutation was found in individuals. Where do they reside from, or where do they reside? So this led to Allison proposing what he called the malaria hypothesis. The reason why this mutation, which is not necessarily good to have, why it persists in the population, is that there must be some selective advantage to having it. And that selective advantage is to fight or combat malarial infections. The malaria hypothesis is a hypothesis that, yes, getting a mutation in beta globin gene caused a phenotype in which some of the red blood cells were misshapen. But that is advantageous for people who are living in an area where there's a parasite trying to affect their blood. So because you are a diploid individual, you have 2 copies of all your genes, one from mom and one from dad. If there is a mutant form of that gene in the population, then you can be an individual who inherits 2 normal copies of the gene, which is probably what most of us in this room are. There's no selective advantage for us to have ever acquired this because we don't experience malaria. But in Africa, where malaria is prevalent at least, this is not an advantageous genotype to have. You're much better off having inherited a single mutant gene and a normal gene, because if you do that, you are producing blood that is half sickled, which causes no disease symptoms, but it additionally provides you the benefit of reduced malaria infection. The consequence of having this in the population is that some individuals will unfortunately inherit 2 copies. Therefore, all of their blood is subject to sickling, which will confer them probably more reduced malarial infection, but at such a tremendously high cost. Weakness, anemia, pain, fever, organ damage, they probably don't live to reproduce. So the evolutionary cost is you will produce fewer individuals that have this, a lot of individuals that have the heterozygous condition, and this is a more fit individual in Africa. In Oregon, this is the more fit individual. So not only does mutation happen, but the pressures that act on that mutation are different in different places. And they determine whether a mutation will persist in a population or whether it does not persist. And so this is a really great example. The heterozygote, which is the one who has the combination of both, has the greatest fitness in Africa. The heterozygote does not have the greater fitness in Salem, Oregon. And so why is this relevant? Well, sickle cell, the mutant sickle cell gene, is prevalent in a population of citizens of the United States, notably those who have arrived from Africa most recently, mostly as a consequence of the slave trade. So this mutation is prevalent within the African American community. And there are a significant portion, somewhere around 11 to 12,000 citizens that have sickle cell disease and that carry the allele. Now, the assumption is eventually, over time, that allele will disappear from the African American population in the United States, but it will likely persist in the population in Africa. So not only is mutation important, but where you are, the selective pressures in your environment determine whether it will persist in the population or whether it will be lost. So that's one example, a classic example of evolution in human, or at least the evidence for evolution in humans. Are there any questions about sickle cell disease or sickle cell anemia? Okay, the second example that I want to talk about is the evolution of lactase persistence. So, how many individuals in this room are lactose intolerant? You'll know, because if you're not sure, just go home and have a big glass of straight milk, and you'll find out quickly enough. So lactose is a sugar, carbohydrate, that is abundant in milk, the milk of mammals. Lactose is processed by an enzyme called lactase. What lactase does to lactose is it cuts it. It cuts it into 2 smaller molecules, galactose and glucose. And it does so because these smaller forms of the molecule can be readily absorbed by your body. Lactose is too large to be absorbed. So an individual who is lactose tolerant, this is the small intestine, Here comes lactose into the small intestine from someone who's had a glass of milk. Lactase is present on the cells that line the small intestine. Lactase will cleave lactose into its two constituent parts, and the small intestine will absorb them. An individual who is lactose intolerant, which in general are all adult mammals. All adult mammals should naturally be lactose intolerant. You're lactose tolerant when you are an infant, when you acquire milk from your mother and you need to break that lactose down to absorb it. When you get older, milk is not the common source of energy. So adults typically do not have the ability to digest lactose because as adults, they stop producing the enzyme lactase. So what happens to an individual that is lactose intolerant? Lactose moves through the small intestine, undigested, into the large intestine where it encounters bacteria. Bacteria are more than happy to digest the lactose themselves, and in the process they produce gas and acids, which results in bloating, discomfort, flatulence, abdominal pain. You'll never touch another glass of milk again. There is a population of individuals, adults, who are lactose tolerant. Somehow they have bucked the trend. They are supposed to be lactose intolerant because as adults, mammals stop producing the enzyme lactase. That's a normal part of mammalian development. Somehow there are lucky individuals, adults who are lactose tolerant. They keep producing lactase through their entire life. These individuals are said to be lactase persistent, meaning they keep producing it. They persistently keep producing it. So how did this happen? Why are some individuals lactose tolerant as adults and other individuals are lactose intolerant? This is another example of human evolution. That there are adults who keep producing lactase throughout their entire life. And it's been traced to a mutation, not in the actual gene lactase, but in a piece of DNA next to the gene that is responsible for throwing the gene lactase on or off. So that might be a little difficult to understand, but think of it like this. If a light bulb in a room is a gene, and that light bulb can be on or off, and you go into a room and turn on a switch and the light bulb doesn't come on, what is your first thought? The bulb is burnt out. That is equivalent to a mutation in the gene. But what if the bulb isn't burnt out? The switch is broken. The light is still off. And so what's happened here is individuals who are supposed to be turning off their lactase gene when they reach a certain point, they have acquired a mutation in that switch that keeps the switch always on. So they produce lactase through their entire life. They have acquired lactase persistent. So aging is a multimodal process. There are multiple genes for aging. Can I answer your question afterwards? There's multiple genes, and they were mostly discovered, by the way, in the fruit fly. So it's a really great organism for understanding humans. But the deal here is that unlike sickle cell disease, the mutation did not occur in the actual part of the gene that makes the protein. It occurred in this, quote, junk DNA that doesn't make protein, but is responsible for switching the gene on or off. And it arose in two different populations independently. So the Maasai, they are lactase persistent because they have a mutation. Europeans from the Central Balkans, Central Europe, they are lactase persistent because they have a mutation that keeps producing lactase. Those two mutations occurred independently of one another, but they were both selected for and maintained in the population because both of these groups were pastoralists. They were highly dependent on their culture of maintaining cattle and raising cattle. So it was a beneficial mutation for them to acquire. This is an example of the coevolution of not two different genes in a predator and a prey, but this is a gene that is evolving because the evolutionary pressure acting on it is the culture of that group. So what is the selective advantage of having or being lactase persistent? The reason I like this example is because it parallels sickle cell disease, but one, it occurs not in the gene itself, but in a different part of the gene, a switch that controls the gene. And second, it arose independently twice. So Mother Nature spontaneously found a way to make lactase persistence not only happen, but stick in two distinct populations on the planet. And the selective pressure to maintain that was obviously strong. So if you are lactase persistent, then you can trace your ancestry to one of these two original cohorts that evolved this trait tens of thousands of years ago. Okay, so those are the two examples that I wanted to present about human evolution. Somehow we seem to think that as humans we are beyond evolutionary change. And the fact is we don't necessarily see it because for us time is the... But I can assure you that there are all sorts of environmental and evolutionary pressures that you experience and that your DNA undergoes mutation and you transfer that DNA to your progeny. So there is no doubt that we are constantly undergoing evolution. We just don't get to stick around long enough to see the end result. We see what's already happened.
Jason, thank you very much. We're out of time and won't have time for questions, but you rose to the occasion. We'd like to invite you back regularly.
Sure.
Thank you.
So, thank you for the invitation. I would just like to make one announcement. One of the things I do in the Department of Biology is I am the director of an imaging facility that we have. So we have a lot of microscopes, electron microscopes. We have acquired this $750,000 laser microscope that allows our students and our staff to take absolutely stunning images in fluorescence. So this is the nervous system of a fly. This is part of the retina. You can see all the little cones and rods here. We have an art show, or is it a science show? We don't know. Is it science? Is it art? That we put on once a year. And the images that students take are absolutely amazing. I would encourage you to come by Olin Science Center. It's on Winter Street. If you would like to ever see the facility or see students using the scopes, they are always down there. They're always working, and there's nothing better than that they love doing than showing off. So if you're interested, they would love to have you drop by. So just drop me an email and let me know, and thank you very much.